So here we are at the National Center for Biotechnology Information website, NCBI. This is a repository where the Human Genome Project basically houses information about the human genome. Now, the word genome includes all of the DNA sequences in an entire organism's DNA set. So all chromosomes in the human DNA set, which includes these 22, which are the somatic chromosomes, and then these two, which are the sex chromosomes, X and Y, and then the mitochondrial genome, which is really tiny. We'll talk about that later in the course. But here you can see all of the chromosomes right here in their ideograms. You can see the salmon color here represents the centromere, and some of these, of course, are metacentric chromosomes, some are submetacentric, and so on, like we saw before. What I want to do is to show you how this is organized and show you some of the information. In particular, I want to go, suppose I'm interested in a particular gene, and the gene I'm interested in is this. It's called the ACE2 or ACE2 receptor. The reason I chose this gene is because it's recently been shown in a paper that was just published in the last few weeks that the coronavirus, the SARS-CoV-2 virus that we're dealing with right now, uses this ACE2 receptor as a way of entering the cells in the infection cycle. We're going to talk about that. We'll show you that later, but I'm curious about this particular receptor right now. So what I do is I type it into the search human genes box here and hit search. And it's going to find this gene for me and any other gene associated with it and put it up on this table right here. You see right there, the very first one is ACE2. So it's got all this information here with all these different genes. Each one of these things is a particular gene. VEGF-A, for example, is associated with this and so on. These are genes that could interact with it in some way or other. But the gene I'm after is this. And I'm going to click on it, and it'll take me to the page for that particular gene. And it does this for all human genes that are known. Okay? So, first of all, it gives you the name, angiotensin 1 converting enzyme 2. That is the name of this particular receptor, and angiotensin converting enzyme is an enzyme that's involved in controlling blood pressure, but this particular thing is associated with receiving that signal, and it gives you that information here under the summary. Now if we go down here a little bit, genomic context will tell you where this gene is, and notice it's on location XP22.2, right? Just as we went over before, that tells you it's on the X chromosome on the short arm in region 2, band 2, subband 2. Exon count we'll talk about later, and it gives you a bunch of information here. This picture here shows you where it sits relative to other genes. So there's VEGF-D, which is associated with angiogenesis, making new blood vessels, and a number of other genes here. It's right in the middle of that cluster. Now if we scroll down a little bit further, we get to this picture here, which looks extremely complicated, and it kind of is. It holds an enormous amount of information about how this gene actually functions and what it actually does. Now, if we go here, though, if I look at this, you'll see there's a slider right here. I'm going to take this slider and crank it all the way to the right so that I'm zoomed maximally. And now look what I see here. I'm right in the middle of the gene, and you see here this upper strand. There's T, C, G, C, A, G, A, and then notice the strand below it is the complement. And notice that right there says 5'. Here's the 5' here. So there's your anti-parallelism. The bottom strand is running 5 to 3' on the left, and the top strand is running 5 to 3 on from the right. So this is what I mean when I say that we've sequenced the DNA. We know the exact DNA sequence of the human genome. But of course you're probably wondering, well, wait a minute, we're all different. So whose genome are we looking at here? Well, the genome sequence that you're looking at here is not any single person's genome necessarily. What you're looking at here is what we call the consensus sequence. Every single base that you see here, there's a TA pair, there's a CG pair, GC pair, AT pair. Each one of those is, in a sense, the most common that's found in that exact location among all the people sampled. And the people sampled here, if we look around, it'll tell you exactly how many people this data set is based on. I don't know what it is. At this point, it's not worth trying to find. But it's going to be on the order of thousands, hundreds of thousands, or even millions of people for this particular sequence. And probably it's closer to hundreds of thousands because, again, this is a very, very important gene. So this has a whole bunch of other information, though, associated with it. This is just the consensus sequence. But follow the cursor down. Now you see there's a C here in this upper strand. If you follow the cursor down, it goes to this red box. That red box is showing you human variation. Of all those people that they sampled, they found that there is, not everybody has a C right here. Some people have an A. And so if you look here and you click on it, it will give you information of exactly what it is that it's looking at. And if you just scroll over it, it'll give you this little box that tells you what it is. It gives you an accession number right here, the actual number for this particular variation ID, basically this particular mutation. It's a single nucleotide variant of length 1, and there are 2 different possible alleles. The majority of people have a C, but some people that they found have an A there. Now notice these regions here, from this to this, there's no red box beneath it. That means every single person that they've ever tested has the exact same sequence. Like for example there, every single person of the hundreds of thousands or however many people that they've tested has a G. Everybody has an A here. So you see, only where there's red boxes have they found any kind of variance. So this is the human genome. And what can you do with this information? Well, this is something that we're going to find. If I can scroll to the right or left, I can move this around and look at the entire gene. And if I don't want to see it in that much detail, I can always just sort of scroll out like this. And you can see as I move it out, you get these red boxes, these red lines with arrows. What are those telling us? Those are telling us how it is that this gene is read and in the direction that it's read. So for example, right here, it's being read from from left to right, which means it's reading this lower strand, because remember, the DNA is built always from 5' to 3'. It's also read always in exactly the same direction, from 5' to 3'. So this particular, what we call reading frame, is moving from left to right. That means it's reading this lower strand here. These down here are different reading frames. For example, this is a gene called BMX. It's the exact same sequence of DNA, but it's a different gene, and in this case, it's reading the upper strand from right to left. So the question that I want to deal with right now is, what in the world is in here? How is the information actually coded, and what information is actually here? That's what I want to look at next. Okay, so what I want to do is show you one gene, one particular gene, and I'm going to show you its anatomy. Now, the part I'm going to show you again is sort of the coding region in just one particular part of its control sequences. The other parts of its control sequences are scattered across the genome, like I mentioned. So this is kind of an older view of it. This is what we would have called the classical gene, whereas the modern gene concept has changed a little bit. Now, when we represent a human gene, it's really difficult to do that on this kind of format. And the reason is the genes are huge. There are thousands of nucleotides or even tens of thousands of nucleotides. And I can't put that on this slide, or else every single letter would just be too tiny to actually see. So what I'm going to do is I'm going to represent only one of the 2 strands. Now, like we saw before, there were 2 different strands there running anti-parallel to each other. I'm only going to show one. And by convention, when I only show one, I'm always going to represent the strand as it's being read from 5' to 3'. So remember, the DNA is always read 5' to 3'. Here's an example. So suppose we have a sequence of DNA that's this, 3' G, T, G, T, blah, 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 all the way to 5'. Its complement, as you know, must run 5' to 3' because of antiparallelism. This nucleotide in its complement is going to be an A, this one's going to be a C, and so on. So we have this complementary nucleotide sequence. This would be the DNA chunk that I want to look at. But if I'm representing it on a piece of paper, I'm not going to write all this because there's a lot of it that's redundant, and it's already hard enough to keep track of these giant genes. So instead of writing all of that, I'm only going to write this strand here, which I call the sense strand, or which is called the sense strand. It's called the sense strand because this is exactly how the gene is going to be read. It's going to be read ACA, ATG, CTT, and so on, not TGT, TTA, because that would be reading in the wrong direction. Not 3' to 5', but 5' to 3. So when I write it as it's understood. ACA, ATG, CTT, GGA, just like this. Now notice then, I don't have to put a 5' right there. It's understood that there is a 5' on this end on the left and a 3' on the right. So whenever we write the gene, that's how we always write it, 5' to 3', and we always write the gene as it's being read by the cell. Okay, so let's take a look at a gene. This is one particular human gene called the beta-globin gene. The human beta-globin gene is a gene that produces a protein that's involved in carrying oxygen in the blood. That protein is complexed with other proteins to make a molecule called hemoglobin, as you know, and hemoglobin is what carries the oxygen. Now, this is just one part of the hemoglobin. It's not all of hemoglobin, it's just one part. So what I'm going to do is I'm going to write the sense strand only. Like I said before, this is the 5' end here, and there's the 3' end. And I cannot represent all of the entire gene on one slide, like I said. It's just too much. There's too much there, and I'll show you what a full gene actually looks like before long. But what we have here then is sections that I've cut out. And when I've cut out sections, I'm just going to put them as dashes like this. And these are sections. They're collections of nucleotides, A, C, G's, and T's, and so on. But they're not anything that I need to focus on right now for understanding the structure of the gene. So each one of these sections is at least 20 bases long, but this particular gene is much, much longer than I'm going to show you. Here's the rest of it with most of it cut out, the vast majority of it cut out. Only the specific sequences that I want to focus on will be shown. Okay, so what does all this mean? This is an actual gene, and if you don't know anything about how to read it, it just looks like a bunch of random letters. In fact, when we first were sequencing or first seeing how these things work, that's kind of what it looked like. We had no idea how to read it. But since that 1953 paper was published by Watson Crick, we've learned a lot about how this information is actually packaged. Now, first of all, this whole thing is the gene, goes all the way to this section here. You notice this section does not look at all random. AAT, a bunch of A's, followed by CAT, and then 2 T's, an A, and a bunch of T's. That sequence you're going to come to recognize means we're looking at the end of a gene. Genes end with that typical sequence. 2 A's, a T, a bunch of A's, 3 random letters, 2 T's, an A, and a bunch of T's. All right, but there's more here. In fact, you can't see it by just looking at the nucleotides, but by mapping how these things operate when we mutate them. Specifically, we've learned that this section up here, from this G all the way to here, to this endpoint, which I'll highlight here in green, is a control sequence. It doesn't have the recipe itself. What it has is a control sequence that tells the cell, when should I actually use this gene? When should I make this protein? And this whole region then that's green, this control sequence, we call the promoter. It promotes the gene. And here's the point. Not all genes are operating in every single human cell. In fact, we have the exact same genes in all different cells. And if you remember, there's about 200 different kinds of cells in the human body. So it's not that different cell types like neurons and renal cells have different genes. They have the same genes. It's just that the genes in a neuron that are turned on are different than the set of genes in the renal cell, the kidney cell, that are turned on. This promoter tells the cell under what conditions should you turn this gene on. This promoter says to this gene, turn this gene on when you are developing into a red blood cell or a muscle cell, or a few others. There's some other possibilities as well, but those are the two main ones. If you're a hepatocyte, a liver cell, this promoter has the information that says don't turn on. Only turn on when you're in those other kinds of developing tissues, especially red blood cells and muscle cells. So all that information is there, and I'm not going to teach you how to read all of that. That will come, and you probably not till you get to graduate school. But there's where the information is, and it's important to know that the promoter then is a control sequence, and it always is at the beginning or ahead of the coding region. There are other control sequences called enhancers, which are the ones that are spread all over the place. This particular one, which we call a cis element, CIS, is right next to the coding region. Now there's a couple of different sequences here that are important. This sequence, CATAAA, that sequence right there is a key sequence in promoters. About half of all promoters have a sequence like this. Usually though, this first letter is not C but T. So it's typically called a TATA box, T-A-T-A box. And it usually reads T-A-T-A-A-A. That particular sequence is the sequence that attracts the copy machine. If you remember, when we were looking at that little story about the crab puffs and all of that, there was the kid in the library who had to make the copy. In reality, it's like a copy machine binds to the gene and then makes the copy of the gene into messenger RNA. The place where the copy machine binds is right here on this TATA box. Now, like I said, about half of all human genes have a TATA box. The other half don't. They have different sequences, a different sequence, but they perform the same function. It's an attractive element that attracts the copy machine. We'll learn what that copy machine is later. All right, so that's what the TATA box is all about. And then if we look here, there's this sequence, CACATTT. That sequence itself doesn't mean anything. However, the copy machine, when it binds to the data box, has a nose section that goes all the way out here and binds right to that A. And when it starts making the copy, it starts right on the A, right there. There's nothing special about the A. It's simply that that's where the copy actually starts. And so the copy begins, or the copy for the gene begins on this A. So the rest of the promoter is not copied, but this portion of the promoter is copied. All right, so that then is something we're going to come back to a number of times, but now I want to go into the next section. Where's the actual recipe itself? The recipe starts right here with this sequence, ATG. That's where the sequence that we call the coding region. The coding region is the exact recipe for how to build this particular structure, in this case a protein. Now, what we're looking at here is the primary structure of the protein. If you remember back from the first unit, primary structure of a protein is the sequence of amino acids. This is where that sequence of amino acids is laid down. It starts here with this ATG. And every coding region of every human gene starts with the exact same 3 letters, ATG. In fact, every coding region of every animal and plant and all multicellular organisms start with ATG. In fact, the vast majority of single-cell organisms also start with ATG. There's only a few weird bacteria that have a starting sequence that is something other than ATG. But right there is where we actually start with the actual coding region that tells the cell what the amino acid sequence is going to be. And it goes all the way through here and continues on until you get to this sequence right here, TAA. That TAA is the end of the coding region. So the coding region starts here, and then it ends there. That's not the end of the gene, that's the end of the coding region. So inside this coding region is the sequence of nucleotides that tells the cell what is the sequence of amino acids. That's the central dogma. That's how the central dogma works. So we're going to come back to that concept in more detail here shortly. But there's something weird about this. The interesting thing about this particular gene is that if you mutate certain of these nucleotides, like for example, if you mutate that C and make it into a G, you get a different, slightly different sequence of amino acids. So if you go to other regions, however, you can mutate the gene, you can mutate the nucleotides like crazy, however you want, and it won't change the structure. And if you map those out, we get this. The yellow sequence, the yellow parts here are the parts where if you actually mutate those nucleotides, you can change the amino acid sequence. But if you go to these red sections and mutate these, it has no effect on the amino acid sequence. In fact, you can cut them out completely and will still have no effect on the amino acid sequence. So this mapping maps out in the entire coding region 2 different kinds of sections. The yellow ones, which are what we call the expressed sections or exons, and these red ones, which are the unexpressed sections. They're unexpressed in the sense that they don't code for particular amino acids. They don't contribute any information to the sequence of amino acids in the primary structure of the protein. So we have 2 different sections in the coding region. The exons are expressed, the introns are not. Now this pattern is true in essentially all eukaryotes, not prokaryotes. The prokaryotes don't tend to have these introns and exons, at least the bacteria don't. So this then is an organizational weirdness of all eukaryotic genes. They have these exons and introns in them. And if you remember, when I showed you the ACE2 gene, it talked about the number of exons and the number of introns. Now this beta globin gene is relatively simple. It only has 3 exons and 2 introns. In that other gene, I think, if I remember correctly, there was something like 11 exons. So the genes can get far more complicated than the one shown here. And then finally, the last section is this purple. The last section, which again goes through this sequence, which I started with, AAT, AAA, 3 weird ones, TTA, and a bunch of Ts. That is the end of the gene. So from here to here is the actual end of the gene. So just in summary, the gene starts here with the promoter. The promoter then gives way to the first exon of the coding region, goes to a series of exons and introns until it gets to the end of the coding region. And that coding region end sequence here is TAA for this particular gene. And then the gene continues on into this section until we get to this bunches of A's, AAT, bunch of A's, 3 weird ones, TTA, bunch of T's. So the question is, why do we have these introns and exons? And what do the introns do, or do they do anything? Well, like we've seen before, and you see this in the history of science quite a bit. When we don't know what it is that something does, we tend to say it does nothing. So I was originally taught, when I was first taught this, we didn't know what the introns did. We didn't know their significance. So I was taught they were garbage. They were just useless, unexpressed nonsense. And it turns out that that is absolutely not true. They do perform a very important function, which we'll get to later on. But the key thing is that for our purposes right now, the exons are the most important parts of the genes in the sense that they hold the information on how to build this primary structure, the sequence of amino acids, as I mentioned before. But what's strange is this. Even though the exons are what hold the recipe, the actual amino acid sequence, they typically only make up about a third of the coding region. Most genes, on average, have about 2/3 intron, which is the unexpressed garbage, and 1/3 exon. So when I was first learning this, again, we were told, well, the genes have all this garbage in it, and it's very, very inefficiently packaged, and it's not a very useful thing, and so forth. Well, it turns out we just didn't understand what was going on, and it should have told us the fact that 2/3 of the coding region was intron should have been some sort of indication that these things are not useless. Natural selection doesn't work that way, typically. If something is useless, there's a benefit to get rid of it. And these things have not been gotten rid of. So we need to understand exactly what those things do. We'll talk about that later, but before we get to that, I want to get a little bit more detail into exactly what that sequence is doing and how it fits into the central dogma.